tissue injury and intravascular thrombosis. Produced with the assistance of the National Heart Institute, the National Institutes of Health, Bethesda, Maryland. Produced by Dr. Philip N. Sawyer, Department of Surgery, State University of New York, Downstate Medical Center. And Dr. Roman Vishniac, Graduate School of Science, Yeshiva University, New York, New York. With the assistance of Dr. E. E. Suckling and Dr. S. A. Wesolowski, State University of New York, Downstate Medical Center. During measurement of vascular transmural potential differences, it was noted that reversal of the normal potential difference across the blood vessel wall frequently indicated the beginning of intravascular occlusion. Because of this finding, experiments were conducted applying electric currents across large and then smaller blood vessels until it was shown that currents as small as 5 to 20 microamperes, currents in the physiological range, passed across the mesoappendix would cause occlusion of the majority of vessels in the tissues. It is well known that injury to muscle tissues results in the generation of a flow of current, known as the current of injury. The question arises, does the spontaneous current of injury in tissues result in occlusion of blood vessels nearby in a fashion analogous to occlusion produced by small applied electric currents? An experiment was designed to measure the effect of an injury current generated by damaged muscle on the blood vessels in a superimposed mesoappendix, an adjacent but separate test tissue. In addition to Dr. Sawyer, who is preparing the experiment, Dr. Vishniac is arranging the cinemicroscopic apparatus, and Dr. Wesolowski is assisting Dr. Sawyer. 1% gelatin Ringer's solution at 37 degrees centigrade is used to moisten the mesoappendix of the rat, which has been placed over a transilluminated lucite rod in the rat board. Controlled experiments consist of this simple arrangement. Here one sees the transilluminated lucite tube over which the mesoappendix will be draped. The cecum and its attached mesoappendix is extracted through the low midline incision in the rat abdomen. The entire cecum is draped with cotton saturated with Ringer's solution. Now the Ringer's drip is started. In the experiments, all visible blood vessels in the prepared mesentery are periodically scanned and given a numerical grade to designate the stage of occlusion. Under normal conditions, all the vessels in the field have high velocity flow. There is some minimal sticking of white cells to the wall of the large vessel in this field. Here is another view of normal flow. Occasionally one sees a cell or two attached to the vessel wall, but the attachment does not take place for an extended period of time. In the left upper corner of this field, Note the capillaries flowing through the fatty tissue and around the fat cells. A piece of rat rectus abdominis muscle is being excised in preparation for the current of injury experiment. The excised muscle, the test object in this experiment, is placed on a piece of cellophane located over the end of the transilluminated lucite tube. Next, the mesoappendix is draped over the injured dying muscle. Ordinarily, we measure the maximum injury potential generated by this injured bit of muscle using saline-filled glass microelectrode pipettes prior to superimposing the mesoappendix. All visible blood vessels in the field are scanned every 10 minutes and their stage of occlusion recorded. Here an artery is flowing over the edge of underlying injured muscle. Cells begin to attach to the vessel wall. Accumulation occurs on the interior of the blood vessel to give the impression that thickening of the endothelium is beginning. The flow is fairly normal. At high power, evidence of rouleau formation is visible. At low power, flow within blood vessels lying over the cut edge of muscle continues, but is decreased. However, the vessels parallel to and closely adjacent to the left edge of the muscle are occluded. Advanced rouleau formation is seen. Apparent endothelial thickening is now very conspicuous. Almost no normal flow is visible. The small vessels are, for the most part, completely thrombosed. Note the embolic phenomenon caused by flow of red cell rouleau with interspersed fibrin particles visible in the smaller vessels. Here is an even more advanced degree of occlusion. A completely occluded vessel is visible, situated parallel to the injured muscle at the bottom of this picture. If one boils the test muscle prior to placing it beneath the mesoappendix, it loses its vascular occlusive effect, as well as its spontaneous injury potential difference. Skeletal muscle, after being macerated and injected beneath the mesoappendix, also has a markedly reduced occlusive effect. Thromboplastin, as used in this experiment, has little of the rapid occlusive effects of the injured living muscle. Grade 3 to grade 4 thrombosis is present in all of the vessels running parallel to this injured underlying muscle in the right upper corner of the scene. The vessels running parallel to and most closely approximated to the muscle are in the majority completely occluded. There is no capillary flow. In fact, the only flow seen appears arterial. Sludging and rouleau formation are seen in all of these vessels 10 to 20 minutes after applying the mesoappendix to the underlying injured muscle. If one drops lactate on vessels which show this degree of occlusion, one can frequently cause dilatation of these vessels and communicating capillaries which are not thrombosed. Rapid streaming of blood through dilated vessels occurs as a response to the deposition of lactate on the mesentery. Two vessels are shown flowing over the edge of muscle, the upper one flowing in, the lower one flowing out. Note the difference in the rate of blood flow between these two vessels of equal size. The one flowing out shows apparent endothelial swelling with slowed blood flow. All of the cells flowing out through the lower vessel show rouleau formation. Many cells are attached and stuck to the blood vessel wall. The blood flowing in across the muscle shows much less evidence of rouleau formation and narrowed lumen. This would seem to indicate that the current of injury produced by the injured muscle causes an electrotactic effect on blood flowing over the muscle surface. The opaque underlying muscle mass prevents visualization of an electrotactic effect on the vessels located over the muscle. Therefore, the closest visible occlusion points are at the edge of the muscle. Note that there is minimal residual capillary flow. This is a picture of the transilluminated mesentery with the opaque underlying injured muscle beneath and the Ringer's solution drip used to keep the preparation moistened. If one places a piece of 5 millimicron pore dialysis membrane between the muscle and the mesentery, this does not appreciably decrease the rate of thrombosis of the overlying vessels. This indicates that the occlusive effect is not produced by large peptides and proteins diffusing across the muscle-mesenteric interface, since the effect continues through the small pores of the interposed membrane. Evidence in this direction is also obtained by the minimal effect of thromboplastin and macerated muscle on mesoappendix blood vessel flow. Here is the end stage of two vessels, one flowing out, one flowing in across the muscle. The vessel flowing in still shows some flow, in spite of the fact that the companion vessel shows complete occlusion with minimal pulsations of the blood clot within the vessel. At higher power, note even in the vessel of entry the advanced rouleau formation, endothelial swelling, and white blood cells sticking to the walls, quite akin to those effects caused by applied electric currents. Large-size emboli stream through the vessel. No capillaries are seen. They are all occluded. Here is more evidence of advanced grade 2 to grade 3 occlusion in a large vein running out over the edge of muscle. Again, the rouleau formation, thickened wall, and fairly advanced occlusion are first visible at the point where the vein can be seen running across the edge of underlying skeletal muscle. Another view of the muscle mesentery preparation and a partially occluded three-cornered branching. When first seen, blood was flowing from the right branch out through both branches of the Y facing to the left. Now the branch on the right is completely occluded by fibrin and cellular clot. Blood flow has changed in that it comes down from the vascular branch above and flows out below. A final view of this vessel 30 minutes later shows it completely occluded. Occasional pulsations of the thrombus within the vessel are produced by the changes in blood pressure caused by the heartbeat. It should be emphasized at this time that these changes are not seen in controlled experiments. Through the capability of Dr. Vishniac's cinemicroscopic experience, we have been able to demonstrate visually for the first time a relationship between biological currents of injury and intravascular thrombosis. The effect appears directly related to the currents of injury produced in the injured tissues. Tissue injury and intravascular thrombosis. Produced by Dr. Philip N. Sawyer, Department of Surgery, State University of New York, Downstate Medical Center, and Dr. Roman Vishniac, Graduate School of Science, Yeshiva University, New York, New York, with the assistance of Dr. E. E. Suckling and Dr. S. A. Wesolowski, State University of New York, Downstate Medical Center.